Today I'm going to talk to you about the work that I've been doing looking at characterising the cell composition and landscape of malignant pleural effusion in metastatic breast cancer. And I'm starting off with the acknowledgement slide here because this work has fundamentally been a fantastic collaboration with Delphine Marino and her team at the Olivia Newton-John and Belinda Yeo, the clinician who we've been working with to collect these samples. In my group, Holly Whitfield, an extremely talented young PhD student, has been doing the analysis that I'm going to present today. I always want to start with that just in case we run out of time at the end. This is work that could not have happened without Delphine and Jean and Belinda. So the samples that I'm going to be describing to you today were collected through the Olivia Newton-John Cancer Research Institute's Breast Cancer Biobank. This is a program that's collecting a large number of samples from both treated and treatment-naive patients, and as well as sequencing a number of these samples, they're also being used for organoid studies to generate PDXs, and we're collecting clinical follow-up as we go. What I'm going to talk to you about today is some single-cell RNA sequencing that we've done on a number of malignant pleural effusions from breast cancer patients across a number of different hormone receptor subtypes. So we have 2 patients from a triple negative subtype, and we have 3 patients who are ER positive, PR negative, and 2 patients who are ER positive and weakly PR positive as well. So we set out to address the question, what cell types are present in malignant pleural effusion? What's the immune component that infiltrates this liquid accumulation in the pleura, and what kind of diversity do we see in the cancer cells? Really looking at asking the question of how well does malignant pleural effusion capture the biology of what's going on with the patient's cancer. We use a number of bioinformatics methods, which I will shamelessly gloss over here for this audience, but we use a reference-based cell type analysis method called single R to try and characterise the cell composition of the sample that we've collected, and a number of other classification methods for looking at identifying cancer cells and the molecular phenotypes of cancer cells. So this is the data set that we have collected across our 7 patients. We've collected about 50,000 cells following filtering, so this is actually quite a large data set. We have around about 30,000 reads per cell across all of our patients, and are measuring somewhere in the vicinity of 18,000 genes in each cell that's been collected. Here I've shown all of these cells coloured by patient, and so you can see in this t-SNE plot there are a number of clusters that purely correspond to single patients, but other clusters that are composed of cells from multiple patients. Now, if we actually break this down and start looking at the cell types that are present, we see a really large range of cell types that have been captured in the pleural effusion. Some samples show very high levels of cancer cells, and others are substantially immune-related, largely cytotoxic cells. And let me take you through the colouring here. On the left-hand side, I have coloured this plot based on patient sample. So in other words, colour corresponds to patient. On the right, colour corresponds to this cell type. So here we can see these 4 clusters, which are purely drawn from single patients, correspond to epithelial cells that we propose are cancer cells. On the other hand, we see a number of these other cell types that relate to immune cells and different immune composition that are a mix of things from different patients. I'm actually just seeing the chat popping up here, so I hope everything's going okay.
Yes, sorry, just making some jokes being made as a bit of byplay. Carry on.
Excellent. Well, in that case, I'm glad it's not a whole bunch of people saying we can't hear Melissa. What has happened to the audio? So if we have a look a little bit closer at the breakdown per patient, what we can see is that only 4 of our patients actually have a substantial epithelial component. So you can see here these 2 triple negative patients, one of our ER positive patients and one of our ER PR positive patients. Each have this large collection of pink cells that are captured and correspond to these 4 clusters here. The other 3 patients have very few, if any, detectable putative cancer cells in their sample, and so don't really show their own clusters across on the side here. You can also see the very large collections of T cells and NK cells, for example, in some of these patients, indicating a very high level of immune involvement. Now, we also have a number of enriched samples that correspond to some of these non-enriched samples. So these are a subset of the pleural effusion in which Miltenyi enrichment was used to effectively deplete the sample of any immune component. So it's a cocktail of antibodies that removes putative non-cancer cells from the mix. And if we have a look at the 3 patients for whom we have both enriched and non-enriched samples, you can see for patient 20 and 66, where we started off with a very large number of cancer cells in the initial sample, we end up with almost purely cancer cells after Miltenyi enrichment. For patient 21, there were very, very few detectable cancer cells in the unenriched sample that we had, but after enrichment, over half of the cells that have been sequenced correspond to a cancer subtype, although we do still see quite a large number of non-cancer cells in those samples. Now, this means that bulk sequencing on an enriched sample would, in the vast majority of cases, we think, yield a transcriptome that corresponds to the cancer of the patient rather than immune infiltration into the malignant pleural effusion. If we look at the immune component in a little bit more detail, again, my matched C-SNEEZ plots have been coloured on the left by patient and on the right by cell type. And you can see here that the clustering is more predominantly driven by cell type. We have a large cluster up here corresponding to monocytes and macrophages. We have a very distinct B cell cluster here, which is composed of B cells from all of our patients, NK cells in pale blue, and the dark purple you can see here corresponds to T cells. If we have a look at that breakdown again, you can see that there are varying proportions across all of the patients. Some patients seem to have much more NK involvement, that's that blue portion there. Others have much more involvement from monocytes, macrophages, or T cells. So it is really quite variable across our patients. If we light these up by biomarker, you can see again that the corresponding clusters show strong expression of biomarkers known to be associated with macrophages, for example. This CD79A is a B cell marker. It lights up that little B cell population quite beautifully. KLFR1 is thought to be a good marker to separate NK cells from T cells. And again, it's highlighting this region through here where we have a strong representation of NK cells. So the cell type identification for the immune component seems to have worked very well. If we look, on the other hand, at the non-immune cells and perform the same analysis, you can see coloured by patient on the left and coloured by cell type on the right, we really see 2 dominant types of cells that emerge after we computationally remove the immune cells. So here we have clusters corresponding to patients, but the pink portion is the epithelial cells, and this orange portion you can see across here corresponds to smooth muscle cells and fibroblasts. Now, we used a copy number analysis approach to try and verify that the epithelial cells that we had identified are indeed cancer cells, and that the CAFs and the smooth muscle cells and fibroblasts are not actually cancer cells. So for this, we used a method called infer-CNV, which takes the single cell RNA sequencing data and performs an inference of copy number based on that. We need to compare to a reference population again. So here we've used a reference population drawn from this paper that I've cited at the bottom here, where we've sampled cells from benign mammary epithelium across 4 different patients. We've also included the immune cells from our own patients in this study so that we have an example of their normal genome to compare to. And what you can see is that this yellow cluster through here corresponds to the cells that we have labelled as epithelial cells, and indeed they have pretty messed up looking genomes when it comes to copy number. Whereas this portion down here are the ones that correspond to the things that we're referring to as CAFs, so fibroblasts and smooth muscle cells. And you can see that these cells are certainly not subject to the same kind of amplifications and deletions that we've seen in the putative cancer cells. So we're quite confident in assigning these cells through here as cancer cells, and these ones down here are likely not cancer but fibroblasts. Now, we identified a population that we think are mesothelial cells in those fibroblasts and cancer and smooth muscle cells. And so we were quite interested in seeing what the potential role for these might be. And this is still work in progress, but what we're doing is using the cell-phone database collection of ligands and receptors to identify molecules that have been secreted by those fibroblasts and mesothelioid cells that have interacting receptors on the cancer cells in our study. So here the size of the node corresponds to the number of cells or proportion of cells that express that gene. And the ones in blue are the receptors, whereas the ones in pink are the ligands. And what we can see is that there are a number of signaling pathways where there are receptors expressed on the cancer cells, things like EGFR and a number of elements of TNF signaling pathway, for example, where ligands appear to be being secreted by cancer-associated fibroblasts and mesothelioid cells. Some of these nodes that have got dark circles around them also indicate cells that, genes that are known to play a role in formation of blood vessels and vascular mimicry. So we think that there may be a role for these fibroblasts in that as well within the malignant pleural effusion. We can also look at heterogeneity in the molecular phenotypes of the cancer cell population. So here now I'm only looking at those epithelial cells from the analysis. And what we can see if we look at the expression of particular markers like fermentin, for example, or CD44, you can see that there's quite a lot of heterogeneity across the cell populations. Indeed, only in one of our patients do we identify a cluster that's really strongly expressing fermentin. We have 3 patients here that have got quite high levels of CD44 being expressed. If we look at Ki67 as a marker of proliferation, we can see that even though we identify quite a lot of these cancer cells across the patients, there are varying degrees of expression of Ki67, indicating that some of these cells might not be proliferating, whereas others might be more rapidly proliferating. We can also apply the subtype classification methods to clustered pseudo-bolt data from the cells across these patients to look at whether or not there's any apparent heterogeneity in terms of the subtype. And so here we're looking at both enriched and non-enriched samples where we have them. The colours correspond to the main subtypes: basal, HER2-like, luminal A, luminal B, and normal-like. And what you can see is that even though these patients have been classified with a particular subtype based on the primary tumour of that patient, you can see some heterogeneity in terms of how subtypes would be assigned based on the PAM50 classifier. What's quite interesting is where we have matched samples and we can compare the unenriched, the enriched sample. For patient 66, it's made no difference. They've all just been labelled as basal. But for patient 20, between the non-enriched and the enriched samples, we see a substantial change in the proportion of luminal A versus luminal B-like cells that have been detected, indicating that the enrichment process itself might be doing something to the heterogeneity that's retained in the enriched sample. So this is going to bear more investigation. We have a look into what's going on with that. So that brings me to the end of that whirlwind tour through the molecular and cellular landscape of malignant pleural effusion. I'd just like to acknowledge the people both in my group and in our collaborating institutions that have been involved in this work. I've already mentioned Holly, who has done a lot of the beautiful bioinformatics analysis here, and my colleagues Delphine, Jean and Belinda over here with Jean, who collected and sequenced the samples. And this is 2020's version of a lab photo. That's my marvellous team captured on Zoom, because that's the only way we get together anymore. So I will leave it at that, and hopefully we still have time for a couple of questions.